This video will look at the diversity of animals. What are some of the basic characteristics of animals? They're eukaryotic. So remember, on our evolutionary tree, everybody in blue and red here is prokaryotic, everybody else is eukaryotic. So they're part of that eukaryotic group. They're also multicellular. So unlike most of the protists, which are unicellular, the animals, fungi, and plants are multicellular. They are also heterotrophic, so that separates them from the plants. Animals and fungi are heterotrophic. So what splits those two groups is how they get their energy. Animals ingest their food rather than absorbing it outside the body. Most animals also have specialized tissues. What are some of the key features in animal evolution? What are some of those major events that occur in animal evolution? One is the evolution of body symmetry. So we typically start off with organisms with no symmetry, then we have radial symmetry, and then bilateral symmetry. With bilateral symmetry, we also see the evolution of increasing cephalization, meaning a head region. We also see major changes in the embryonic germ layers. We start off with organisms with no germ layers, then we have 2 germ layers called diploblastic, and then 3 germ layers, which is called triploblastic. We also see the evolution of a coelom, which is a body cavity. We start off with organisms that have no coelom, acoelomate. Then we get the evolution of a false coelom, pseudocoelomate. And then we get the evolution of a true coelom, the eucoelomates. We also see differences in the way the gut forms during embryological development. So we have 2 different groups here. First we have the evolution of protostomes, and then later we have the evolution of deuterostomes. Another thing that occurs with gut formation through evolution is we go from an incomplete digestive tract to a complete digestive tract. Incomplete only has one opening. So one opening serves the function of both the mouth and the anus. In a complete digestive tract, we have that one-way flow of food from the mouth to the anus. Let's take a look at each of these. First, let's look at symmetry. We see on the far left, we have organisms that are asymmetric, like sponges. In the middle, we have the evolution of radial symmetry. So if I make any cut, as long as I go through the middle of that organism, I will get equal parts. Bilateral symmetry on the far right, there's only one way I can make that cut to get equal parts. I can get an equal left and right. I can't get an equal front and back. I have to make it down that midline. When we look at the embryonic germ layers, we first start off with organisms that are diploblastic. They have 2 germ layers. That's represented here. The digestive cavity is in the middle, endoderm around that, and then on the outside in blue, we have ectoderm. So we have endo and ectoderm. In our triploblastic organisms, we get the addition of a 3rd germ layer, which is this red layer in the middle called the mesoderm. When we look at coelom formation, we start off with organisms that are acoelomate on the far left here. They have no coelom. A coelom is a body cavity. If you look at the organs inside your body, they are inside of a cavity. So there's a space around those organs. It's not right up against your muscle wall. In an acoelomate, it is. So we have the digestive cavity, the lining of the digestive cavity, and then right up against that is muscle tissue. Then we also have pseudocoelomates on the far right. They have a coelom, but it's lined on one side with mesoderm and on the other side with endoderm. Here's that coelom. We see endoderm on the inside and we see mesoderm on the outside. If I look at eucoelomates, we have the coelom here, which are these little pouches that form, but then we have mesoderm all the way around it. If we look at embryological formation of the gut and the coelom, it can happen in 2 different ways. Both of these organisms are eucoelomates. How that coelom in the gut develops is a little bit different. And it's different in 2 ways. It's different in where the origin of the mouth is. Embryologically, when that mouth forms, it forms in different places in protostomes and deuterostomes. And it also is different in how the coelom forms. Now let's place those characteristics on a phylogenetic tree of the animal. We have the animals. Some have no true tissues, like the sponges. The rest of them all have true tissues. The evolutionary origin of true tissues occurred here. After we get the formation of true tissues, we get the formation of symmetry. So down here we have organisms that are radially symmetric, and then up here we have organisms that are bilaterally symmetric. After we get the formation of this bilateral symmetry within that group, we get the formation of the coelom. This group down here has no coelom, everybody else has a coelom. Once we get the formation of a coelom, we split them into protostomes and deuterostomes. These two groups down here are deuterostome. Everybody else up here is protostome. In this protostome group, we also have a major split in the ecdysozoa. These are organisms that form a cuticle or an outer exoskeleton that they shed. And then we have the lophotrochozoa that do not do that. They go through a common type of embryological development. Let's take a brief look at the animal groups. First, we'll look at the sponges. They have no true tissues. So everybody else that's in the animal group has true tissues, but the sponges do not. So they have cells that perform certain functions, but not true tissues. This shows us the basic structure of a sponge. Water will come in through these ostia into this cavity in the center of the sponge called the spongiocele, and then out through this opening here, which is the osculum. Other characteristics are cells called choanocytes, a layer called the mesohyl, spicules in some, and they also have amoebocytes. This is that inner spongiocele. There's the ostia. These are these choanocytes. They are the collar cells. And if we look at that choanocyte in a little bit bigger view, it has this flagella, has a little collar around it. This creates a current which moves water through the sponge and also brings food particles this way back to these amoebocytes, which will consume those food particles. These amoebocytes are here next to the choanocytes, and they are in a sort of a gelatinous material called the mesohyl. And then often we will see spicules forming which solidify that body. Next, we'll look at the cnidaria. They are radially symmetric organisms. Some of the key features of cnidaria, they have two distinct body plans. They have the medusa that we see there on the left, which looks kind of like a jellyfish, and then we see the polyp over there on the right, looks a little bit more like a sea anemone. They have two tissue layers. Have an epidermis and a gastrodermis. You can see up here the epidermis is in red and the blue is the gastrodermis. In between, we see that gray area represents a jelly-like substance that is known as the mesoglia. It's not cellular in nature, so it's not a true mesoderm. That's why we call it mesoglia. They also have a gastrovascular cavity. That's a single opening. Food particles come in and waste particles go out that one single opening. Because it not only is a gastric cavity, it also serves for their vascular system. That's why we call it a gastrovascular cavity. They also are characterized by having specialized cells called nematocysts. Nematocysts are stinging cells. So a jellyfish stings using these nematocysts. The nematocysts may have poisons in them. They may have barbs. Here we see some of the cnidarian diversity. Cnidaria include the sea anemones, jellyfish. We have fish in quotations there because they are obviously not truly fish. And then we have the hydra, and we also have box jellies. Next, we'll look at the platyhelminthes, which are the flatworm. We see them over here on our phylogenetic tree. They're bilaterally symmetric. I can cut this animal down the middle and get two equal left and right parts. Organisms that have bilateral symmetry are also triploblastic. They have 3 germ layers. And we can see that back here on our phylogenetic tree. These organisms are acoelomate. So that's a little bit confusing when we look at our phylogenetic tree because we think the acoelomates are down here, but then we have the platyhelminthes, which are acoelomate, and they're up here in organisms that should have a coelom. The reason for this is that based on other characteristics of this group, the evidence shows that their ancestors probably had a coelom and then this group has lost it. They also have an incomplete digestive system. There's one opening we see here that serves for the mouth and the anus. Excretory system essentially regulates water and nitrogen waste, which in us is urine. They have this network of tubules that runs throughout the body for that excretory system. They have a rudimentary nervous system. They have 2 nerve cords that runs down, almost creating sort of like a ladder down the body. They also have sensory cells at the front end, so there is some cephalization, the formation of a head, and they have their sensory system concentrated in the head ends. They have these eye spots, so they are sensitive to light. They also have chemosensory cells, which allows them to respond to chemicals. Notice they have no circulatory system. They do not have a respiratory system. These are small organisms that live in aquatic environments. They have moist bodies, and so they exchange gases across their body surface, and they do not have a circulatory system that then transports those gases. This looks at some of the diversity of our platyhelminthes. So we have our planarians, which are free-living. The other groups are parasitic flukes. There's two different types of flukes, the trematodes and the monogenea. And then we also have the tapeworms. Next, we have the roundworms. These are nematodes. We see the nematodes up here in the protostome group and in the ecdysozoa. They are pseudocoelomates. We now have a complete digestive tract, so we have a separate mouth and an anus. So we have that one-way flow of food. One distinction is they have a hard cuticle made of chitin on the outside of the body. And that's what puts them in this ecdysozoa group, because in order for them to grow, they have to shed that cuticle. Next, we have the arthropods. The arthropods are also ecdysozoans because they have an exoskeleton that they have to shed in order to grow. This is the largest phylum of all, contains the most species. They have jointed legs. That's what arthropod means. They also have segmented bodies. Their exoskeleton is made of chitin. They have a true coelom. They also have a hemocoel. What they mean by the hemocoel is there's a cavity that the blood flows into. In our bodies, the blood is contained within vessels throughout its entire route. Organisms like this, which have what we call an open circulatory system, there's a group of beating hearts that push the fluids, but it goes into this large cavity. They are protostomes, ecdysozoans. They have this exoskeleton that has to be shed in order to grow. Arthropods have a wide variety of respiratory systems. Some of this depends on whether we're looking at a terrestrial or an aquatic group. On the left, we see that arachnids have structures called book lungs, which looks like layers of a book. That is where oxygen and CO2 exchange occurs. In our crustaceans, we have book gills. They are very similar to book lungs, but they're external to the body. Because this organism is in water, it can have those external. You can't have external lung surfaces in an organism that is terrestrial because a respiratory system has to be wet. And if you had this wet respiratory system on the outside of your body, it would just dry out. Insects are a little bit different. They have a tracheal system. Tubes run through here that carry the air. And then we also have openings to the outside in these little holes called spiracles. Here on the side of this grasshopper, you see these black dots that go down the side. Those are the spiracles. The majority of the air going in and out is occurring down here through these openings on the abdomen. We look at arthropod diversity. We can see the major groups. We have the trilobites, which is our extinct group. We have chelicerates, the myriapods, the crustaceans, and the insects. This trilobite belongs to an extinct group of arthropods. If we look at the trilobite, we can see that it has a lot of body segments. If we look at insects, we can see that these body segments are fused into three major regions. We have a head, and we have a thorax, and we have an abdomen. So we have fewer body segments than we saw in the ancestor. They have 6 legs that are attached to the thorax. So all of the legs come off of this middle region here. If there are wings present, they are also attached to the thorax. They have a complete digestive system, so we have a mouth and an anus. They use that tracheal respiratory system. They have a very well-developed nervous system with strong centralization, so that means that we have sort of a brain region. When we talk about it being centralized, it means a lot of the function are in one location as opposed to being spread throughout the body. They will have one pair of antennae. They also have very well-developed sensory systems. Next, we'll look at the myriapods. This includes the millipedes and centipedes. They have many legs and many body segments. They also have one pair of antennae. Here we see a centipede. Centipedes have one pair of legs coming off of each segment. So if we look at the segments, we'll see one pair of legs coming off of each segment. These are carnivorous and venomous. So you don't want to mess around with centipedes. Millipedes, if we look at each body segment, there's actually 2 pairs of legs. These organisms feed more on decaying matter, roots, leaves. They are not venomous. They're not carnivorous. Now we'll look at the crustaceans. One difference we see, say, from the crustaceans to the insects is that we have what's called a cephalothorax. So we actually had fusion of the head and the thorax region into one large area. It's often surrounded by a carapace. You see the carapace in this bottom diagram. They have a heart in the dorsal thorax region here. Many of them will have very hard exoskeletons, often harder than a lot of the insects because there's a lot of calcium carbonate embedded into that exoskeleton. And this group includes the crayfish, lobster, shrimp, crab, and barnacles. Next, we have the chelicerates. The distinction with chelicerates is their first set of appendages is modified into a structure called a chelicerae. This group includes scorpions, spiders, ticks, mites, and horseshoe crabs.